So in our final video on photosynthesis, we can finally do the other end of photosynthesis. We talked about the light-dependent reactions. Now we're going to talk about the other side of it, the carbon fixation reactions. And we'll finish with this one last flowchart called carbon fixation reactions. And now we don't want to technically call it the light-independent reactions simply because we're going to be utilizing and requiring certain products. It requires the carbon fixation reactions, requires things like ATP and also NADPH. Absolutely must have these two things, and these two things are going to eventually form or help form, let me rewrite that, help form carbs. Specifically carbs from CO2, carbon dioxide. So once again, you don't want to call carbon fixation light independent reactions because you're requiring things that come from light reactions. And then you can't necessarily be specifically, technically considered completely light independent because you need things that come from light. ATP and NADPH, we proved in our past 5 or 6 videos that are started and are sort of sparked by the light energy of photons. But overall, let's get into the idea of carbon fixation now. Carbon fixation means, you know how you fix lunch for yourself or you fix a sandwich? You're making a sandwich. What you're doing here is making carbs from carbon dioxide, and we'll see how we do that. And by carbs, I mean simple sugars like glucose, fructose, whatever the plant needs at that time. In addition, the overall chemical reaction of this process is 6 CO2 plus 12 NADH. All of these are coming from the environment, and the NADH and 18 ATP that I'm writing here are coming from where? They're coming from the light-dependent reactions. This CO2 is coming from us. We breathe this out, shows us the relationship that we have between plants. So we input these products, and we get this as reactants. We get as we input these reactants, and get these products. We get 6 molecules of H2O. We also get 12 molecules of NADP. Not NADPH, and then we get 18 molecules of ADP and 18 molecules of phosphate, inorganic phosphate. So that's our overall chemical reaction. We can refer to that as we continue down this line. This overall process, carbon fixation, happens utilizing a cycle known as the Calvin cycle. Now this is, again, the beauty of the idea between respiration and photosynthesis being almost exactly the same. Like, we had the Krebs cycle in respiration, right? Now we have another cycle, and that Krebs cycle, remember, it occurred where? The mitochondrial matrix. This Calvin cycle is actually, it happens in almost all plants, some form of it at least, happens in most plants. And it happens in almost exactly the same region of the chloroplast instead of the mitochondria, called the stroma. The stroma is basically the chloroplast matrix. And it's amazing to me how similar the two processes are going to be as we do the cycle on the side over here. The stroma, it happens, that's where it happens. In addition, there's actually no direct use of light. Notice the terminology, no direct use of light energy. That's why some people call it light-independent reactions, because there's no direct use. Technically, these Calvin cycle reactions can happen without light, because if you have ATP and NADPH, you can undergo the Calvin cycle. But again, indirectly, you get ATP and NADPH from light, so you can't necessarily call it completely independent. These both, again, like I just mentioned, involve ATP and NADPH. They are musts. They are inputs that must be there. They are energy molecules that will energize and provide energy for this cycle to occur. There are about 13 reactions that occur in this cycle, and there are 3 phases. Those 3 phases include carbon fixation. That's one major part of this cycle, carbon fixation. They also include reduction phase. We'll go over each of these. Very difficult to write so small. Reduction phase. And then the last phase is called RU, not Rutgers, but RUBP regeneration. And I'm just going to write regen just to save space, because we're going to draw a nice big cycle here. So what we're going to first do is carbon fixation. That's our first step. And what's going to happen is I'm going to start with, imagine I start with CO2. That's what we start with. we have 3 molecules of CO2, let's say. I start with 3 molecules of CO2. Keep in mind how many molecules I'm starting with and what I'm going to be doing. So 3 molecules of carbon dioxide, that's a 1-carbon compound, and I'm going to combine it with 3 molecules of RuBP. That's a compound that actually has 5 carbons. So if I combine a 1-carbon carbon dioxide compound with a 5-carbon compound, I'm going to end up with, overall, a product. Let me actually draw that over. I'm going to end up with a product over here. And this is going to be our step 1. And our step 1 is actually catalyzed by an enzyme protein known as Rubisco. Rubisco is the most abundant protein in the world. It's an amazing fact because every single plant, plants are everywhere, utilizes this first step. Rubisco is the most abundant protein in the world. Just a fun fact for you. So we combine carbon dioxide and RuBP to create 3 molecules of, I'll just say, an unstable intermediate. And if you combine something that's 5 carbons and 1 carbon, you've now created a 6-carbon molecule, right? So if we've created a 6-carbon molecule and it's unstable, and I told you that it splits, if it splits, you expect it to turn into not 3 molecules now. If I split these 3 molecules, I've now created 6 molecules. Because you have, let's imagine, 1, 2, 3 unstable molecules, and now they're all going to split in half. If they all split in half, you've created 1, 2, 3, 4, 5, 6 total molecules that are now going to actually have a name. That name is 1,3,6,1,3 biphosphoglycerol. GLY, we'll just say. Again, the names aren't incredibly important, but just keep in mind the amount of molecules that we're using and the steps that we're undertaking. So that was step 1. Step 2 is going into a normal state, a more stable compound known as 1,3-biphosphoglycerate. Oops. This is actually supposed to be not 1,3-bisphosphoglycerate. Sorry about that. This is actually called 3-phosphoglycerate. Sorry about that. Phospho-GLY. Okay? Same thing. Everything's still the same. But this is now a 3-carbon compound. Once we've created this 3-carbon compound in step 2, we're going to now undergo step 3. Step 3 involves ATP. You need to use ATP. You're actually going to use 6 molecules of ATP to create 6 molecules of ADP. So you're going to take 6 phosphate molecules and attach them onto this structure. And you still have 6 molecules overall of this new compound, this 3-carbon compound, but instead you're now actually going to have a new compound called 1,3, this is where it comes in, bisphosphoglycerate. Because what have you done? You've just added another phosphate group. You first were phospho, now you're biphospho, because you've just added 6 additional phosphate groups to these 6 molecules, creating 6 1,3-biphosphoglycerate molecules. These are all still 3-carbon molecules. So we have a 1 carbon plus 5 carbon giving us a 6-carbon molecule that immediately turns into a 3-carbon molecule that turns into still a 3-carbon molecule, but this time has phosphate groups on both the 1 side and the 3 side of the carbons. So now, moving forward in step 4, so that was step 3. In step 4, what we have is the utilization of NADPH. We're going to take NADPH, which is again a high energy component, and turn it into NADP. But this is going to catalyze a reaction that's going to change this completely into 6 molecules of what is known as G3P. These are still 3-carbon compounds, but G3P is something you've seen before. Again, this shows the interconnectedness between us and plants. G3P is a very high yield molecule. It's a molecule that's very, very important because what's going to happen is 5 out of the 6 G3P molecules will go back into the cycle. They'll just continue the cycle as is, so 3-carbon molecules still. But one of them, where happened to the one? What happened to that one that was left out? One G3P molecule will make the bold decision of actually leaving the cycle and actually turning into eventually glucose. This is how, ladies and gentlemen, plants make glucose. They utilize the ATP and the NADPH specifically 6 molecules, sorry, six of the light-dependent reactions to turn carbon dioxide into G3P. Once they've turned carbon dioxide through these 4 steps into G3P, G3P will leave the cycle, only one molecule at a time, will leave the cycle and turn into glucose, will turn into food for the cell, for the plant. But the 5 molecules that go back into the cycle are now going to actually turn into 3 molecules of RUBP. And once they turn into these 3 molecules of RUBP, there are actually now 5 carbon molecules. And guess what those are going to do? They're going to go right back into where they started. Look at the connection, look at the cycle that just was created. This is the Calvin cycle. I just went from our starting component of carbon dioxide and RUBP, and I went back to RUBP and still have, let's say, another 3 carbon dioxide molecules waiting for me on the side that'll come in once these guys are gone. So overall, we've done the Calvin cycle. And specifically, I missed one thing. At this step right here, we actually utilized 3 ATP molecules to create 3 ADP molecules. So we're inputting ATP to create 3 RUBP molecules. Overall, I want to label the steps now of carbon fixation, reduction, and RUBP generation. Of course, right over here is RUBP generation. Going from, let's say, step 4, this is step 5, and step 6. So this area right here is RUBP generation. Regeneration, at least. So right here is RUBP generation. Carbon fixation happens all the way up to step 4. So steps 1 through 4 we can label as carbon fixation. And then steps 4 through 5, let's say, 4 through 5 is the reduction phase of the Calvin cycle. Overall, what I want to really emphasize and sort of close off with is this idea that we've gone from carbon dioxide utilizing ATP and NADPH and fixed it and made glucose because of this process. Don't worry so much about the intermediates. Don't worry so much about the naming conventions. Just realize that carbon dioxide combines with this molecule called RUBP. Know that Rubisco is the initial catalyst. That initial catalyst will turn it into an unstable intermediate. That unstable intermediate will turn into sort of 2 stable intermediates. Through this process of carbon fixation, which involves energy, ATP and NADPH, we create a very highly touted, highly revered molecule of G3P, 6 of them. For every 6 molecules of G3P, 5 of them will actually go back into the cycle, and only 1 of them will turn into glucose. This is highly inefficient. It's very inefficient to me that to only have this much success rate in terms of creating glucose. When we want to create energy, every single glucose molecule turns into energy, whereas when they want to make glucose, only 1 out of the 6 possible turn into glucose. So that shows sort of the disparity between the abilities of us and plants. Overall, this Calvin cycle, don't get too tripped up on the amount of numbers that are seen here. This is just a nuance. This is Calvin cycle. Imagine it happening in 3 turns. It's a bit confusing, but this is how your textbook presents it. Imagine this only really happens 1 carbon dioxide at a time, and it happens again and again and again. If it happened 3 times, if 3 carbon dioxide molecules went through this cycle, you would get these results, and you would only get 1 glucose molecule for every 3 carbon dioxide molecules that enter the cycle. That's overall the carbon fixation that we see. So overall, the Calvin cycle is considered the part of the light independent reactions, but once again, let's not call it that because they're ATP and NADPH necessary, and those are both from the light-dependent reactions. So we can sort of conclude on this fact. Plants utilize light to turn it into chemical energy. How do we do that? We undergo light-dependent reactions, and then we undergo carbon fixation reactions. We fix carbon, we turn it from carbon dioxide into glucose through these Calvin cycle steps. Know the steps from a very broad perspective and understand the 3 components of fixation, reduction, and regeneration to give us the overall final results of going back to what we started with, and of course glucose coming out in step 5. Hope you have a better understanding of photosynthesis. Hope you appreciate the process a lot more.